We are live now.
Good morning, everyone. Good morning. Welcome back. Welcome back to the Center for One Health's conference for this year. Day two today. We have a 9 o'clock start. So I hope you've all made your cup of tea and we can start. So my name is Georgios Miliotis. I'm a lecturer here for the discipline of bacteriology in NUI Galway, and I will be chairing the morning session, which will run between 9 and 10:30. And the idea behind the session is that we will focus on antimicrobial resistance from an One Health perspective. So without further ado, I would like, with great pleasure, to introduce the first speaker for today, the keynote speaker, Dr. Adrian Muwonge. Dr. Muwonge holds the very prestigious Chancellor's Fellowship at the University of Edinburgh. He's focusing on digital One Health, and he's also the Deputy Director for the Fleming Fund Fellowships at the University of Edinburgh for Uganda, Malawi, and Kenya. And Adrian specializes in molecular epidemiology, and he now develops technology to enable implementation of One Health strategies with a focus in Africa. And his main area of research, of course, is very relevant to this session, is antimicrobial resistance development and dissemination at the human-animal interface. Just before we start, if you have any questions for Adrian or for any other of the speakers, please use the Q&A function at the bottom of your Zoom link. And we're going to run all the sessions. So Adrian will be first, then we'll have 3 early stage researchers giving their presentations. And then at the end, we're going to have a thorough Q&A session. So thank you all for joining this morning. And Adrian, we're ready when you are.
Morning, everyone. I hope you can see my slide. And you can hear me?
Yeah.
Yes. Good morning. First of all, I am honored to be here, present some of the work that I've been doing as part of my BBSRC Future Leader Fellowship that ended in 2020. Really looking at the subsistence farming and the challenges related to antimicrobial resistance. But before I do that, let me spend just a minute or two to talk to you about what I am currently doing as part of the roles that I play. One, with the Fleming Fund, basically what we do there, we are providing capacity building to the 3 countries mentioned, Kenya, Malawi, and Uganda, where I am talking to you from now. We work with ministries of agriculture, Ministry of Health and Environment, and they nominate individuals that we take through training, and the training really is focusing on how they can work in the One Health, using the One Health approach. And we hope that by building this network of individuals, it will be them who will support the implementation of the AMR national action plans in those countries, as well as evaluation of those national action plans within country and between countries. The second bit, which is really an area that I've coined myself, which is Digital One Health, is trying to develop technology that allows the communication between the disciplines that represent One Health, and to be able to equitably share data while respecting the data security aspects that are between these disciplines. And here, I'll talk to you about just two tools that we have been developing. Really, we got some funding and we've been building a tool that provides market intelligence for farmers, specifically in the pig sector in Uganda. And this we are piloting in 5 districts. What this tool does basically is to provide market intelligence, because farmers want to sell whatever they are producing at a good price. And most of the profits usually are siphoned off by middlemen. So having this data-driven type of tool allows them to sell their products at a fair price. But in so doing, we also track the prices of specific antibiotics that are used in these districts to as granular as a sub-county level. The second tool that we're using also in tandem is called Pig Boost. This is really a service delivery tool that tracks service delivery and productivity on farm. And within this tool, we have also incorporated a component that allows us to track the use of antibiotics at individual animal level. In this way, we are able to really understand how antibiotics are used. And the tool in the last 10 months, Pig Boost, the market intelligence tool, has about 16,000 data entries, whilst the POCMAT, which is tracking animals, we have about 18,000 animals. And over the course of the next 3 years, we'll probably have about 50 to 60,000. And this is really a healthy data set that allows us to understand the use of antibiotics in this sector. The other tool that I am really working on, which is really in silico, it's in the virtual world, is using the technology that underpins cryptocurrencies, which is distributed ledger technology, really to understand how antibiotics are distributed, and from that estimate how antibiotics are used in human and animal sector. So these are the tools really that I am working with so far. Now, as part of COVID-19, with the experience we had shown developing tools, we got funding from NIHR and MRC to build a digital contact tracing tool that is tailored to the whole age sector in East Africa. So these are the 4 tools that I'm working with to create this digital One Health space as an area of research. So back into really the understanding of the occupational risk of AMR. In developing countries, subsistence farming contributes a significant proportion of the value chain, even when the intention is really to keep animals for household consumption. There's a significant proportion that is sold that accounts for a large proportion of meat that goes into the value chain. And if resistance is to emerge from the subsistence farming, really it will make its way to the general population through 2 routes: through the value chain, where the product is delivered to the consumer, but also the direct contact and transmission from the farmers. So it is critical for us really to understand the dynamics on the farm, for the sole reason is if we're going to evaluate animal targeted livestock interventions that are geared to reducing resistance in the human population, we really need to understand the risk groups that are likely to introduce this resistance to the general population. We've stepped back a bit and look at resistance. Resistance itself really exists naturally in the environment, but we have sped its emergence and transmission because of our recent activities and how we're using antibiotics. But really within an environment, if there is a selection pressure, for example, that which is caused by the use of antibiotics, you will have microbes selected for and against, and those that are selected for really will dominate. And they dominate, and how they dominate is really through exchanging what makes them resistant between themselves, but also being transmitted across to other hosts. And these really happen in a niche, and a niche really describes the ways in which microbial species use their environment and how they respond to resource distribution with and in competition with others, and ultimately how those organisms really alter the environment for their benefit. And my favorite niche is the gut. Reason being, a gut is really like, a bioreactor that all mammals are walking around with, and it's a highly diverse environment with many factors that affect the physiology, but also the physiology affects the composition and the structure of the microbiome in this niche. Obviously, along the line of the gut, you have different microenvironments that are curated by the pH and all sorts of other factors. So the point I'm trying to make here, if you're going to study the gut, really you have to find ways of accounting for the variation that is caused by many other factors, including genetics, stress, antibiotic use. And the basic study design that I have used really is to look at the gut as an in and out compartment. And basically the inputs here that affect the gut system include the diet, include antibiotic use, they include there are temporal signals really that affect the gut itself, and where you live really also curates the gut microbiome. If we look we're going to look at this in terms of antibiotic resistance. If we take a sample, a fecal sample, for example, which will be a proxy of the microbial environment within the gut, and grow some bugs or some sentinel bacteria, it gives us insight into the microbial resistance within that gut. And that will be really underwritten by a molecular architecture within that gut system. And so if we do this longitudinally, we're able to profile characteristics in the gut of the pig and the gut of the farmer and relate them to antibiotic resistance and see if there are differences really in how these two hosts that are living in close proximity behave with regards to antimicrobial resistance. So we then take this framework and try to put it into the framework of occupational risk, which represents the proportion of being exposed to a hazard and the danger that hazard would really bring to your life as a function of where you work. In this case, the risk is being exposed to resistance that is generated from the livestock, from the subsistence farming system. And this would be a function of the host themselves, where they live, the amount of antibiotics they're exposed to. That will inevitably have an impact on the microbial characteristics. It will have an impact on the selection pressure of the bugs that are in there, and ultimately will lead to all be represented as phenotypic resistance. And whether or not that phenotypic resistance is transmitted across to the farmer or the pig, this is really the occupational risk that I am trying to investigate. And so I've taken this framework and run a longitudinal study in Uganda for 1 year following farmers in urban and rural settings. And the reason why, as to why we use urban and rural settings, the assumption is that the farmers in the urban settings have disposable incomes to buy antibiotics because they're not free. And therefore they would be using more antibiotics than those in the rural setting. So that creates sort of a gradient in selection with regards to antibiotic use. So we followed 35 farmers in the urban, 35 farmers in the rural, and their pigs. And when I say pigs here, I mean sows, because the sows are the ones that spend a longer time on the farm because they're used for breeding purposes. We also included controls, a positive and negative control. Because we're looking at the relationship between the farmer and the pig, we wanted to look at the extremes. A positive control would be an individual who's exposed to the gut content of a pig, frequently. For example, a person who works in the abattoir who eviscerates the gut content of pigs. That would be our positive control. Now, negative control would be a person who would never be in contact with pigs because of religious faith. So these were our controls. So we collected fecal samples and ran a digital, a mobile phone-based questionnaire to collect a whole host of information to account for the variations that we will encounter downstream. And we did this for a year, going back every 2 months. So that would be 6 times for each farm. And so we use this integrated, at least according to me, integrated microbiome approach. The numbers you will see peppered within this figure are the numbers either of samples or of sequences or isolates that were collected. So from a fecal sample, we cultured 2 sentinels, E. coli and Klebsiella, and then we profiled these on phenotypic resistance with 6 antibiotics that are used in human and animals. We also extracted DNA from the same sample and quantified 4 genes that correspond to the antibiotics, some of the antibiotics that are used. These genes include TET-Q and TET-B, which encode resistance for tetracyclines, and EMRB, which encodes resistance for macrolides, and DFRA, which encodes resistance for trimethoprim and sulfamethoxazole. So we quantified these genes in each of the sample across time. We also then submitted the same DNA for 16S sequencing so that we can profile the microbiome based on amplicon sequencing. Obviously, that is cheaper. We could screen all the 968 samples, but for the shotgun metagenomics, we really had to select 100 of these samples, which represented about 8 households in the urban and rural settings and their controls. So this is the framework that we used integrated to really try to characterize the microbiome of a farmer and a pig and try to make sense of what it means with regards to the resistance. So let's start off with the phenotypic resistance as the result. The phenotypic resistance here, on the x-axis, we have the 6 antibiotics that we profiled against, and on the y-axis is the percentage of isolates that were resistant. As you can see, the resistance is highest for trimethoprim, tetracycline, and streptomycin. And generally, the resistance was higher in urban compared to rural settings. The lowest resistance was seen for ciprofloxacin. But in general, you had more resistance for the E. coli than Klebsiella, and the resistance in the controls were significantly lower, both lower than the farmer and their pig. So there is a difference between what is happening on the farm and what is happening elsewhere. And we already see the signal between urban and rural settings. That was monoresistance. If we then look at multi-resistance, on the x-axis, 0 represents the number of antibiotics to which a sentinel is resistant to. So 0 would mean they are susceptible to all of them, and 6 would mean that they are resistant to 6 antibiotics. And that is the profile across the board of these. You can still see that in the urban settings, it was higher. And generally, the profile of the farmer and the pig is really not different. The percentages of resistance between farmer and pig are comparable. But I wanted to draw your attention to the 3 groups that I've highlighted, group 1, group 2, and group 3, because I'll be using them downstream to look at how resistance and phenotypic resistance is related to all the other microbiome factors. And group 1 represents susceptible, and group 3 represents the most resistant. So it provides us a gradient of resistance that we could look at. This figure really is a compression of the entire study. We are looking at households. So on the y-axis, the numbers that you, the IDs you see there is each of the household. And on the x-axis, the first panel really is looking at the antibiotics that we profiled across time. So you can see how the antibiotic resistance at household level has changed. And the reason why I'm showing this is really to show that this resistance is across the board in the urban setting. It's not just specific areas, the entire repertoire of farmers that we followed in the urban settings actually exhibited this level of increase. And when you compare that to the 4 panels to the right, which is quantifying the qPCR genes carried, you see that those farmers will also carry more of these genes than their counterparts in the rural setting. So what about the microbiome characteristics between the farmer and the pig? The panel A, you can really see that the gut microbiome of the pig is far more diverse than that of the farmer. But we can also see that there are differences between the farmer and the controls that we have used. Panel B shows you the comparison between the diversity of the farmer and their pig. And we have colored these by the setting, which is urban and rural. What this shows that you have a higher diversity in pig and farmer in the urban settings than in the rural. What we see in the rural, though, is that there's an inverse relationship between the farmer and the pig with regards to the diversity, and we need to explore what that really means. We then explore further the relationship within the microbiome between the gene count according to qPCR and the diversity that we have. What these figures really show is what we have seen already, that the pig has a higher diversity than the farmer, but we also see that as the diversity increases, the count of genes reduces. This suggests that there is likely a specific group or taxa of microorganisms that are carrying these genes, because if these genes were carried by all the organisms, you would expect that as the diversity increases, that would increase. And this is true for both the farmer and the pig, but that relationship in terms of the correlation is much more clearer when you look at the genes that encode resistance for tetracycline. And if we look at the controls are also, although the diversity is similar or closer to that of the farmer, the amount of genes they carry is lower than what the farmer carries. So there are relationships really with the farmer. And now in this figure, this is when I want to draw you back to those groups that we selected, group 1, group 2, and group 3. What these figures show is that basically the gene count of microbiomes from which you recovered a bacteria that is susceptible to all antibiotics will have a lower gene count, whilst one where you have recovered one that has multi-resistance will have a higher gene count. And that is true across all these genes. And this is not necessarily the case for the controls, but this relationship is actually true for both urban and rural, which is likely to be really a fundamental aspect about this relationship. And so we also then look at how these groups that represent the difference in a phenotypic resistance are associated to the structure of the microbiome. And here we use the figure on figure A. You can clearly see that the farmer and the animals are separated, but within those groups, you can see that the groups, group 1, group 2, and group 3 also cluster which suggests that there's a relationship both in the human and animals with regards to the resistance groups. And figure B really, what it suggests that the diversity really reduces as you have these multi-group, multi-resistance bugs isolated from a microbiome. So basically, a microbiome from which you isolate a sentinel that is susceptible to all antibiotics is more likely to be diverse than obviously that then reduces when you are isolating those that are multi-resistant. So we also then wanted to look at the whole host of other factors that will affect the structure of the microbiome. And here we use the permutative analysis of variances that is used in microbiome analysis. And we use these two distances, the unifrac, weighted and unweighted unifrac. The weighted takes into account the phylogenetic structure. What we see is the temporal signal. Time has obviously an impact on the structure of the gut microbiome, both in the pig and the farm, and the setting where the R really explains a lot of the differences that you will see, but also the amount of the genes that they carry really, specifically DFRA also explains the amount of variation in the structure. And for humans specifically, TET B. And these are sort of indications of what likely how the antibiotics are used between farm and pig. So basically, the factors that affect the structure of resistance in the farmer and their pig are really not any different. And so when we look at the shotgun sequencing data, which really gives us the entire repertoire of resistome, and here I've simply focused on 4 classes of antibiotics and the genes that we were able to retrieve from these shotgun sequencing data. What you see, for example, for beta-lactam, based on the diversity of genes we see, I think these classes of antibiotics are used really at the same frequency in human and their pigs. There might be a difference in the types of antibiotics that are used, but the intensity of use of those is likely to be the same. The macrolides are also likely to be used both in pig and human, although the types here of antibiotics are really likely to be different. And here we really also see the signatures that are really unique to the urban and rural setting as highlighted. For the aminoglycosides, I really think that these are likely to be, there's more use in the pigs than in humans, given the number, given the diversity that you see and the number of individuals from who we recover these genes from. The picture is rather different for tetracycline because the profile is really the same between farmer and pig, and indeed the controls. So this really has to be has to have had ubiquitous use over the years. So if we go back really to this framework that I'm trying to put across in terms of understanding occupational risk of resistance, putting together all these factors to try and explain the phenotypic resistance that we see. And here we have, these are really preliminary results because I haven't fully included the whole range of metadata to try and explain the phenotypic resistance that we see. But in the preliminary results of the model that I've run, the things that I've just demonstrated to you above hold true also in the model, because we are counting for the variance of all the other factors. Here we use a mixed effects logistic regression model, and we're using the household as a random variable. And we see that you're most likely to encounter resistance in the urban setting, and you're less likely to encounter resistance with Klebsiella. Obviously, the resistance is likely to be due to these antibiotics, and you're less likely to. So the farmer, as you encounter, you're more likely to encounter resistance in the farmer than in the pig. The only exception is the urban pig at the bottom, where if we have used an interaction term, we find that the urban pig is really carrying more resistance than the pig in the rural setting. So back to the question, what really is the occupational risk? And this is really work in progress that I'm about to finish. The patterns for me reveal that really there must be a direct relationship, contact or transfer of bugs between farmer and pig. The direction, I couldn't be able to tell you that right now, and that is also work that will come. But based on the profiles of really the fundamental aspects of that would characteristics relating to resistance, I think that that relationship really is likely to be transferring microbes across, and some of them will be carrying resistance. This is likely to happen more in the urban settings. It's very likely that humans are also seeding resistance into the animals in these settings. And just food for thought. I mean, there's this relationship that we've seen regarding the severity of resistance and the diversity of the gut microbiome is something that we could explore or exploit with probiotics, whether or not increasing the diversity of a gut microbiome might get rid of the resistance. That is something that we're yet to explore. So I'd like to thank you for listening to me, but also acknowledge the team that I've worked with throughout the 3 years we've done this piece of work in Uganda, both teams in Edinburgh and teams in Uganda. Thank you for listening to me.
Adrian, thank you very, very much. Thank you for a really, truly wonderful talk. Thank you. So I have a lot of questions, both from myself and from the crowd that has gathered here today. But I'm going to keep them all for the end. I just have one preliminary question before we move on. You've mentioned at the end, and I think that's that's an excellent example of how like a combination of culture independent methods along with bioinformatics and a very rich metadata set can really guide public health advice and showcase this relationship between the environment, or whether it's the environment or the animals and humans. But at the end, you've mentioned probiotics, right? So my only question for now would be, I know for a fact that a big issue with farming is the excessive use of antibiotics. And of course, now there's strict regulations about using them as growth promoters. But in the case of a disease, an animal disease, you still have to resort to antibiotics. So is there a probiotic alternative we could think of maybe? Or to have? I think you're muted, Adrian. Sorry.
We have been doing a bit of work on that aspect and to explore how the use of probiotics really can reduce the number of bugs that are resistant, which in a sense probably would allow us to use fewer antibiotics or less antibiotics in animals, because the bugs are resistant, we tend to use more of the antibiotics. And so this observation for me could really, if this relationship really holds, that having multi-resistant bugs is associated with microbiomes that are less diverse, then probably you would start by adding a probiotic to increase the diversity, to dilute them out, and then give an antibiotic and probably use less of the antibiotic then. I don't know, but it sounds like a logical thing that's worthwhile really looking into.
Okay, and before we move on, just one more question. So what actions should be taken given the kind of the similarity of the resistance patterns that you've observed to mitigate against the occupational risk that you've identified? So what is the suggestion to policymakers, let's say, or what can we make out of what actions can we take to mitigate that risk a little bit?
That's a really loaded question. But what I know from interacting with farmers, and especially interacting with farmers with social scientists and social anthropologists, what we have realized that actually it's a much more complex. The things that drive farmers to use antibiotics are far beyond what we think as scientists. The motivations range from the need to really keep their animals healthy, to preventing disease, to ensuring that the profits are high. And so from a scientific point of view, it's very easy to say, okay, ban the antibiotics or stop them using antibiotics. That's really not going to work. So I think we really have to find a blend of solutions that capture the fundamental science, but also take into account the sociological angle of antibiotic use. And I would be the person to encapsulate all those things.
Oh, that makes perfect sense. Yeah, that's a great answer. Thank you very much, Adrian. Thank you. Okay, so we're moving on now. We'll move on to the first early stage researcher presentation for today. So I would like to introduce Alexandra Schweri, who has been a research assistant here in the discipline of bacteriology since July 2020. Alexandra has a master's degree in biomedical science, and she has previously worked on the One Health EJP WorldCom project, which related to the development of real-time diagnostic on-site tools for the detection of zoonotic pathogens and resistance markers. And Alex will be presenting on this project today. And she's currently a team member of the PEER project here at NUI Galway. So Alex, we're ready when you are.
Perfect. Thanks, Georgios. I'm just going to share my screen here. Perfect. Okay. Good morning, everyone. Georgios, thank you for the introduction. As Georgios said, my name is Alexandra Schweri. I am currently on the PEER project, but today I'll be presenting on the One Health EJP WorldCom project, which I was on from July 2020 until June 2021. The title of my presentation is The Rapid Detection and Discrimination of Closely Related Enterobacteriaceae, CTXM Group 1 variants, BLAS CTXM1 and BLAS CTXM15, using an internally controlled multiplex loop primer endonuclease cleavage, loop-mediated isothermal amplification assay, so a Leclamp assay. Kind of a mouthful, but I promise I will not take up all of your time with the title. So just an introduction to what CTXMs are. So cephalotaxamases are extended spectrum beta-lactamase ESBL enzymes, widely distributed amongst Enterobacteriaceae, especially Escherichia coli. In the context of antimicrobial resistance, these genes confer resistance to third-generation cephalosporin antibiotics. These antibiotics are really good at treating lung infections, such as pneumonia, and they are widely used to treat gram-negative bacterial infections, such as them caused by E. coli. So today I'll be talking about the two CTXM group 1 variants, BLAS CTXM1 and BLAS CTXM15, which are associated with animal and human infection respectively, and are an increasing public health concern, which require rapid and accurate diagnostics. The problem is that CTXM1 and 15 both have highly similar, nearly identical gene sequences. So it's hard to differentiate between the two. Even though it's clearly one is human, one is animal, there are very few single nucleotide polymorphisms or SNPs involved in each sequence. which I'll talk about later. And so distinguishing between these could be vital in investigating a One Health diagnostic approach between human and animal infections. I'm just going to go into a little bit about LAMP and Leclamp. So LAMP is a rapid and cost-efficient diagnostic technique. It's an alternative to PCR. So whereas PCR uses thermocycling, LAMP uses isothermal temperatures and uses 4 to 6 specific primers for 6 to 8 target regions to cycle amplify DNA. Now, similar to PCR, normal LAMP reactions would also require fluorescence. So you would use a dye such as cyber green in normal LAMP reactions. In LAMP, targets are amplified at a constant temperature, so 60 to 65. For the purposes of our experiment, we use 63. So you can pick any range in that. It's just optimization, really. So it avoids the need for a thermocycler, which is required for PCR, making it cost efficient as well as rapid. So today I'll be talking about Leclamp, which is a loop primer endonuclease cleavage LAMP, which is a novel LAMP technique developed by Dr. Owen Higgins at NUI Galway that utilizes a primer probe within a basic site flanked by a fluorophore and quencher that can bind to a target sequence. And cleaving this amplicon at basic site produces the fluorescence. So instead of using a dye such as cyber green, the probe is built with a fluorescent tag. So when that's cleaved, that produces the fluorescence. So when a SNP, or single nucleotide polymorphism, is present in the target, it actually inhibits the cleavage, preventing the fluorescence and enabling the SNP differentiation. So with 2 highly similar sequences, such as CTXM1 and 15, the SNP differentiation could really mean the difference in an important diagnostic tool. This is just a picture of the LAMP mechanism here, so I'll go through this very quickly. So we'll start at the top here where you have your inner primers, your outer primers, and your loop primers. Your inner primers and your outer primers are the 4 basic primers that you would use. And then your 2 loop primers are used in exponential amplification, which then makes up the 6 primers, so 4 to 6. So we'll start at the top here, where the forward inner primer hybridizes to the complement of F2 in the target DNA to initiate complementary strand DNA synthesis. The F3, which is the outer primer, then hybridizes to F3C, so its complement, which initiates the strand displacement synthesis, which releases the complementary strand, which goes to the next step. That complementary strand then serves as a template for the BIP, the backward inner primer, to synthesize a new strand, which is then used as a dumbbell shape. So you can see the 4th one down. So that's what makes LAMP LAMP. That's what makes it loop isothermal amplification. And so then the loop primers down at the bottom, they're what increases your exponential amplification. So they will hybridize to the B2 and F2 respectively. Now this is a mechanism of Leclamp. So Leclamp uses a basic site, a fluorophore, and a quencher. This binds to the LAMP amplicon loop. And so in the wild type one, you can see the black triangle there. That's the endonuclease 4, which cleaves the target bound double-stranded DNA, producing the fluorescence there. Where you can see in the single nucleotide polymorphism, the mutant allele, that the SNP actually blocks fluorescence from being produced. So that way we can differentiate between a target sequence that has a SNP and one that doesn't. So that's what's going to differentiate between 1 and 15 in this circumstance. So overall, the aim of the study was to develop an internally controlled Leclamp assay for the differential detection of 1 and 15 variants in a single reaction and provide a rapid sample prep method that can be used to identify AMR markers portably on site. The detection of these human markers on farms may be indicative of other potential AMR markers that warrant further testing. We are working, there's a consortium that we're working with, and other universities have other target genes that we're looking at, such as OXA48, KPC, and NDM. But we are looking at CTXM for the NUI Galway consort of the project. So the internally controlled Leclamp assay utilizes 2 fluorescent tags, so FAM and hex for 1 and 15 respectively. And then we also have an internal amplification control, an IAC, which has a Psi5 fluorescent tag. This reaction was run at 63, as I stated previously, for 60 minutes. But our results show that the assay time could be reduced to 20 based on our limit of detection studies. Our rapid DNA extraction of porcine fecal samples was achieved using Chelex, a boiling step, and a dilution of the resulting solution in tris EDTA. Our Leclamp assay demonstrated complete analytical specificity of both group 1 CTXM variants using environmental isolates. So these isolates were clinical or from water samples like estuaries from Ireland and central Germany. We also had veterinary samples as well. So they would just come cultured and we would just extract the DNA from there. On our inclusivity panel, we had CTXM1, which had 15 samples, CTXM15, which had 37 samples, and other CTXM variants, so 2, 9, and so on and so forth. And we had 12 of those samples. The assay demonstrated a low-level detection for both CTXM1 and 15. So that was approximately 10 genome copies in approximately 20 minutes. Right now, I'm just going to go through the method that we used for our porcine fecal samples. So we would receive the pig feces and then get a swab the pig feces, put it in an Eppendorf tube of heated Chelex. We would then vortex that for 1 minute and then put it in a thermal shaker at 99 degrees Celsius for 10 to 15 minutes. Another vortex step for 1 minute, a dilution of tris EDTA, and we would just put all of that into the LAMP reaction. So altogether, our method is about 20 minutes, and the resulting LAMP reaction can take up to 20 minutes for our results specifically. So all in all, it's a under 1 hour CTXM 1 and 15 differential Leclamp assay for rapid diagnostics. So now we get to the nitty gritty, our assay performance. So on the left here is our CTXM1, that's in the FAM channel. And on the right is our CTXM15, which is in the hex channel. So as you can see for both assays, we are getting detection of 10 to the 3, 10 to the 2, and 10 to the 1, all before or at 20 minutes. And so our limit of detection right now is 10 genome copies, and that is with optimization as well. And then below we have our IAC, which just makes sure that our assay is working properly. So you can see at the bottom that the no template did not come up, which is always a good thing. And all of our samples came up in a rapid time. Now we move on to our animal sample testing data. So this would be our porcine fecal samples. So the previous were our environmental samples. So this would be our porcine fecal samples that were CTXM positive from Germany. And so on the left, again, we have our CTXM1 because all of the samples were CTXM1 positive. We have the percent of E. coli in each sample resistant to ceftiofur. So ceftiofur is a cephalosporin used in veterinary medicine. So it's just an antibiotic that we're just seeing if it's resistant to. So you can see on the FAM, the CTXM1 graph, that all of the samples came up before 15 minutes. Which was a great thing to see, that our assay was specific enough to pick up up to 25, up to 100% resistant, down to 25% before 15 minutes. And another IAC just to make sure that our assay is still working, which it did, which is always a good sign. So our conclusions is that the development of the CTXM1 and 15 Leclamp assay was successful in differentiating between the two highly similar sequences with 100% specificity and low-level detection of 10 genome copies detected at approximately 20 minutes. Our DNA was successfully extracted and detected from porcine fecal samples using our rapid DNA extraction technique and our Leclamp assay. This is the first reported portable diagnostic technology that can successfully detect and differentiate between 1 and 15. So what makes this such a novel study was that it's portable and can be achieved in less than an hour. So our future directions would be to actually bring our assay out onto farms and do additional evaluation using the portable instrument as a marker for the presence of antibiotic resistance on these farms. So I'd like to acknowledge my colleagues, Dr. Owen Higgins and Dr. Louise O'Connor. The supervisors for my project are Professor Terry Smith and Professor Derville Morris. Our funders are the One Health EJP. I'd like to thank all of the members of the WorldCom Consortium and all of the members of the ARME group for their continued feedback and support. Thank you.
Thank you very much, Alex. Great work. Thank you. It's a great presentation. Yeah.
Thank you.
Brilliant. So we'll keep all the Q&A for the end. So I'll move on if that's okay. So yeah, moving on, we have the next early stage researcher, and that's going to give us a presentation today. So we will be hearing from Maeve Louise Farrell. Maeve is a PhD student here in the discipline of bacteriology. Her research focuses on antimicrobial resistance in bathing waters and aims to better understand if we can pick up antibiotic-resistant bacteria from recreational water activities. And her project, PEER, is being conducted in the Antimicrobial Resistance and Microbial Ecology group here in NUI Galway, and it is funded by the Environmental Protection Agency. So, Maeve, we're ready when you are.
I will just share my screen now. Can you see that okay?
Yeah, we can, yeah.
Perfect, thanks, Georgios. So good afternoon, everyone. As Georgios already said, my name is Maeve Louise, and I'm the PhD student on the PEER project. I'd just like to take this opportunity to thank the organisers for the invitation to speak today, and also thank everybody for tuning in. So I'm just going to begin with a quick breakdown on the PEER project. So PEER stands for the Public Health Impact of Exposure to Antimicrobial Resistance in Recreational Waters. It's a 4-year project funded by the Environmental Protection Agency Ireland, and essentially is a multi-sectorial collaboration between researchers in NUI Galway, HSE, University of Exeter, Queen's University, and ARM Research Group here in NUI. We want to investigate whether regular water users are more at risk of becoming colonised with AMR organisms than people who wouldn't use the water as regularly, so are non-water users. And we want to improve upon existing policies regarding the environmental monitoring of AMR. And I'm going to be talking today about 3 work packages in this project. So work package 1, which is a review on the risk of exposure to waterborne organisms of public health concern in bathing waters, and also work package 2 and 3, which is the recruitment of participants, and the colonisation study, which is to assess the gut colonisation with AMR organisms and also assess the persistence in the gut over a period of time. So just a brief background, I don't need to explain antimicrobial resistance to you anymore, but we all know it's a global public health concern that requires a One Health approach. Currently, there's an approximate 700,000 deaths occurring each year, which amounts to a cost of €1.5 billion per year in the EU alone. Some of the causes of AMR, as we've already heard today, are the overuse and misuse of antibiotics in a clinical setting, alongside the utilisation of antibiotics in both human and veterinary medicine, and the overuse of antibiotics in agricultural settings. Additionally, the lack of and inadequate treatment of wastewater, where occasionally wastewater discharge is released raw into our environmental waters. So carbapenemases are enzymes that are known as beta-lactamases because they inhibit the action of most beta-lactam antibiotics. So these organisms, these carbapenem-resistant organisms alongside ESBLs, as Alexandra explained earlier, are listed in the category priority 3 critical organisms by the World Health Organization for the list of research and development of new antibiotics. And as you can see by the graph here, this shows the EARS-Net data on antimicrobial resistance in Ireland until the first quarter of 2019. So this shows a gradual increase in the multi-drug resistant organisms, and multi-drug resistant organisms would include organisms that are resistant to our 3rd generation cephalosporins, aminoglycosides and fluoroquinolones, so some of our last resort antibiotics. And it shows that there's a need for surveillance of these organisms from a One Health perspective, so not just a clinical setting, but also in an animal sector and an environmental perspective. So we all know that recreational water activities are important for our health and well-being, and they greatly increased during the pandemic. So European bathing season, runs from the 1st of June to the 15th of September each year. And essentially designated bathing waters are sampled just before and during the bathing season for 2 organism groups. So our intestinal enterococci and our Escherichia coli. And essentially they are given a water quality classification from excellent to poor, so excellent, sufficient, excellent, good, sufficient and poor. And these are based over a 4-year period. So when you see bathing water has an excellent status, that would accumulate a total of 16 samples over a 4-year period, so 4 samples per bathing period. In Ireland, there's 148 identified bathing waters in 2020, so the most recent report. And of this, 96% met or exceeded the minimum required standard, so are either sufficient or higher, which is brilliant. Unfortunately, there were 4 bathing waters that were classified as poor, and generally the most common source or issue for these bathing waters was urban wastewater, where urban wastewater is either released in stormwater outflows, so when there's excessive rainwater and plants can't manage the amount of water, or occasionally they are released raw straight into the environmental waters. So to begin with the PEER project, I began a review on the waterborne organisms of public health concern in bathing waters. And from this review, we found that there was a critical lack of surveillance evidence for organisms beyond the scope of the Bathing Water Directive, so beyond our intestinal enterococci and beyond our E. coli. There were 60 studies included in total from a European perspective, and only 27 of these clearly detailed water quality. Of these, 20 described water quality as sufficient or higher. And of these studies, there was 43% of total samples where waterborne organisms of public health concern were detected. So this could have been viral, protozoan, or bacterial beyond our E. coli or our intestinal enterococci. In the AMR category, there was 442 samples that were tested for AMR bacteria with a detection rate just shy of 50%, which is incredibly worrying. And overall, we recommended that there's a harmonization of sample volumes collected and methodologies for analysis so that we can compare, we can expand upon these 16 studies and compare further studies, and also compare them from a global perspective to review the presence of waterborne organisms and also assess the risk. So we decided that there was further research to define suitable indicators that represent 3 categories, so viral, protozoan, and bacterial, alongside risk-based identification of locally important organisms for monitoring. But you can read more about this in our recently published paper in Water Research. So just to go through the recruitment in the PEER project. So we had a national distribution of participants, which were broken down into 2 sampling groups. So our point prevalence study, where participants were invited to submit a fecal sample and survey during a 1-year period. So September 2020 to October 2021. So this is just concluded. And then we will be also doing a longitudinal study, which is essentially a persistence study, including a cohort of the or a subset of the participants from the point prevalence study. And we will follow these people over a 1-year period and collect 6 samples. And just to reiterate that we're looking at 2 specific types of antimicrobial resistance. So our extended spectrum beta-lactamase producing Enterobacteriaceae, ESBLs, and our carbapenemase producing Enterobacteriaceae, so our CPEs. So our recruitment campaign changed dramatically. So when the pandemic came around, originally we had planned to do a face-to-face recruitment strategy, but we swapped to an extensive social media campaign, and that began in September 2020. We used Facebook and Instagram video adverts and also stories to target individuals for inclusion, and targeted the entirety of the Republic of Ireland. And we had a last call recruitment campaign to recruit more non-water users over a period of about 18 days, so the 10th to the 26th of September this year. As you can see on Twitter alone, we earned nearly 10,000 impressions over this period. So you could say that our social media campaign was incredibly successful. So just to break down then the point prevalence recruitment, we had a first cohort where 174 participants were sampled during the September 2020 period to May 2021. So this would be our non-bathing period. And then we had a cohort 2 where 135 individuals returned samples during the first period, so September 2020 to May 2021. And then again, they submitted another sample during the bathing period of May to October of this year. And then 120 individuals also submitted samples during the May to October period of this year. As you can see, we had a good breakdown nationally across all for both non-water users and water users. So just to get into the age distribution, so we had a bell curve show, which was to be expected in line with other studies. For our water users, the 45 to 54 category proved most popular, while with our non-water users, 35 to 44 category proved most popular. Unfortunately, we were lacking individuals in our 18 to 24 age categories, and we did attempt to rectify this by targeting the student population, but we just proved that middle-age groups were more happy to return packs. And then to go through our gender age data, as you can see, it's very obviously female dominated. And with regards to point prevalence 1, we had 86 to 66 for our water user categories of females to males. And then for our non-water user category, we had 92 to 64 females to males. As you can see also in point prevalence 2, it is quite female dominated. And while we did attempt to rectify this also by making sure to choose both female and male participants, females are most likely to return their packs. And briefly, I'm just going to introduce you to the laboratory methods for peer. So essentially, very scientifically, we ask them to provide a grape-sized fecal sample. So we send them out a pack with a survey, and they return this pack with the fecal sample and the survey included in the consent form. Once we have the fecal sample, we streak these directly onto selective chromogenic agars. So our Brilliance ESBL agar, which targets ESBL organisms, and our M. supercarpa agar, which targets CPE organisms. From there, we identify via MALDI-TOF, and we store any Enterobacteriaceae identified at this period. We then subject them to antibiotic susceptibility testing against a panel of 16 antibiotics, and anything that we suspect is an ESBL producer or a CPE producer, we then confirm via real-time PCR. We also hope to do whole genome sequencing in the future and to do more analysis on both fecal samples and the organisms detected. So our ongoing work, our point prevalence results are ongoing with the laboratory analysis hoping to conclude soon, and we're hoping to publish these results in the coming year. Our longitudinal study is going to commence soon, and we're going to assess the persistence of AMR in the human gut, or if people are picking up AMR organisms at specific times of the year. And we hope to aid in the development of monitoring strategies and policy for AMR prevention and control in the environment. And also that we need to hopefully establish a better understanding of risk groups that could be disseminating AMR into the environment, as that was mentioned earlier. I'd just like to take the time to acknowledge the principal investigators, so Professor Dervla Morris and Dr. Liam Burke, our funders, the Irish Environmental Protection Agency, and everybody in the lab, especially Alexandra, who presented earlier, and Dr. Louise O'Connor. Thank you very much.
Thank you very much, Maeve. Very, very informative. Thank you. What a great study. Thank you very much. So now we've already heard from the human-animal interface. Now we've heard from the environment to human interface. So we have covered the whole One Health kind of triangle there. Okay, so the last presentation for this session will be by Niamh. So Niamh Cahill has been working as a research assistant in the discipline of bacteriology since 2017. Her research primarily focuses on antimicrobial resistance in the environment, and she's currently working on an EPA and HSC funded project called ARREST, which is aimed at understanding persistence and transmission of AMR in the environment. So hello, Niamh, we're ready when you are.
Perfect, I'll just share my screen. Okay, so I hope you can all see my screen and hear me okay. Okay, so hi everyone, and thanks Georges for the introduction and for asking me to present or inviting me to present today. So my presentation is on the detection of MCR8 and MCR9 genes in the environment in Ireland. So as you'll have heard from the other speakers in this session, antimicrobial resistance is a global public health concern. It's one that's continuously escalating and limiting treatment options. So colistin is among the last line antibiotics for treatment of infections caused by multi-drug resistant bacteria. It was produced by a soy bacterium and it was first discovered in 1947. So in humans, it was used from 1959 up until 1970. But it was then replaced by alternative antibiotics such as aminoglycosides and beta-lactams due to toxic side effects. But due to increasing levels of multi-drug resistance, it has been reintroduced as a last line antibiotic in later years. In animals, it's a little bit different. So it was extensively used for decades, not only for treatment and prevention of infections, but also for growth promotion. But over the last number of years, it has been relooked at and reevaluated. Its use in animal health is being reevaluated due to the increase in multi-drug resistance and including colistin resistance. So a number of countries have actually banned its use recently. And in Ireland, in April of this year, the Minister for Agriculture, Food and the Marine announced that colistin in animal health use would be discontinued. So just to give a brief background on colistin resistance, so it can be chromosome encoded or plasmid encoded. Up until 2015, all of the reports on colistin resistance were due to chromosomal mutations. However, since 2015, mobile colistin resistance genes have been detected. So these were first detected in 2015, and they were, this was an MCR1. So it was detected in China in humans and animals. Since then, 9 other types of MCR genes have been detected. And today I'm just going to focus on MCR8 and MCR9. So MCR8 was the first detection was reported in 2018, and the first report of MCR9 detection was reported in 2019. Since their detection, they have both been detected in a variety of different bacterial species in human, animal, and environmental samples. So, as we know, a huge emphasis has been placed on the One Health concept, which obviously we know recognizes the link between human, animal, and environmental health. In relation to MCR genes, they have been detected in humans, animals, and environmental sources across the globe. They're therefore highlighting their spread. But when we look at in Ireland, there's no routine surveillance for or reporting of MCR in Ireland at present. And there's very little actually reported on MCR detection in Ireland. MCR1 and MCR1 has been detected in both humans and animals in Ireland. However, there are very limited reports on this. But in relation to the environment, there has been no report of any MCR detection in the environment in Ireland. So as part of the ARREST project, which George has mentioned, is a HSE and EPA funded project that is aimed at understanding the role of the environment in the persistence and transmission of AMR. So as part of this project, a selection of multi-drug resistant isolates had been recovered from environmental samples. and these were sequenced. And it was through the analysis of this sequencing that MCR8 and MCR9 genes were detected. So as I mentioned, the samples were all collected as part of the ARES project, and the MCR8 and MCR9 genes had been detected in samples of freshwater, wastewater, and integrated constructed wetland samples, and these had all been collected between November 2018 and October 2020. So just to point out, the freshwater and wastewater samples were all processed by our group at NUI Galway, and the integrated constructed wetland samples were all processed in the Department of Agriculture. So just to briefly run through our methodology. So initially samples were collected, they were processed, and bacterial culture was carried out. And then bacterial identification was carried out on suspect colonies from the agar plates. So we were looking for Enterobacterales, which are a family of gram-negative bacteria, and we use MALDI-TOF to identify these. So antibiotic susceptibility testing was then carried out on these particular isolates, and then isolates underwent real-time PCR for the detection of carbapenemase and ESBL genes. I won't go through the real-time PCR results today because I will be focusing on the whole genome sequencing results. So a selection of isolates were sent for short-read sequencing, and then a variety of different tools were used for the analysis of this data, which I will mention again shortly. So then any of our isolates that were positive for MCR genes, they underwent Cliston MIC broth microdilution testing. So this was basically to determine if they were resistant or susceptible to this particular antibiotic. So overall, 8 isolates were detected. 8 MCR positive isolates were detected. So 1 MCR8 and 7 MCR9 positive isolates. And these were detected in 6 different samples. So a freshwater lake sample, a freshwater river sample, a hospital and long-term care facility wastewater sample, a hospital wastewater sample, wastewater treatment plant influent, and ICW influent, which is our integrated constructed wetland. So overall, all of our samples were collected from different locations, except for these 2 samples that I've highlighted here. Although they were collected within the same vicinity, they were collected from different sewers, and as you can see, on different dates, so 1 in 2018 and 1 in 2020. And although they contained waste from the same hospital, one of the samples also contained waste from the long-term care facility. So a variety of different species were detected. So these are all Enterobacterales species, and they included Klebsiella, E. coli, Enterobacter, and Rotella. And they were found to belong to a variety of different sequence types. So we characterize these using MLST, and a few of these have been detected previously in humans in Ireland, in hospital sinks in the UK, and in pigs in the USA, and in humans in Egypt. So I know I had mentioned that MCR genes hadn't been detected or haven't been really reported. MCR8 hadn't been reported in Ireland previously. So this is actually unpublished data, and we were informed by the National CPE Reference Lab in Ireland that they had detected an MCR8 positive Klebsiella pneumonia that also matched the same sequence type as our environmental isolate in a clinical sample this year. So they've allowed us to include this for comparison purposes in our study. So these are the phenotypic results then. So these are the antibiotic susceptibility testing results. So these are antibiotics across here that we tested for susceptibility to, and then we have our isolates listed here. So the red boxes indicate resistance to the antimicrobial agent, and green indicate susceptibility. So overall, the majority of our isolates were multi-drug resistant, which means that they are resistant to 3 or more antimicrobial classes. So the isolates demonstrated phenotypic resistance to aminoglycosides, to beta-lactams, to quinolones and fluoroquinolones, to chloramphenicol, trimethoprim, tetracycline, and Cliston. So just due to the interest of time today, I'm just going to focus on the Cliston antibiotic susceptibility testing results. So as you can see here, the MCR8 positive Klebsiella pneumonia was resistant to the Cliston antibiotic. However, all of our MCR9 positive isolates remain susceptible. So this has actually been reported as well in the majority of the literature, that I have come across in relation to MCR9 presence so far. So a lot of them tend to report that MCR, in the presence of MCR9, susceptibility to colistin remained. However, some studies have indicated that it is inducible, so it may just be in the absence of a suitable inducer, the gene isn't being expressed. So these are the genotypic results then. So I know there's a lot on this slide, so I'll just go through the main findings with you. So these are the antibiotic resistance genes here in the MCR8 isolates. And as I mentioned, I've included the clinical MCR8 just for comparison purposes. And then we have the MCR9 isolates down here and all of the genes. So the red boxes indicate that the gene was not detected and the green indicate that it was detected. So we used ResFinder and CARD databases through Abricate to determine the antibiotic resistance genes. So in the clinical isolate, you can see that there were 20 antimicrobial resistance genes detected overall. And then in the MCR8 positive environmental isolate, you can see that there were 25 genes detected overall. In relation to our MCR9 isolates, the number ranged from 11 to 60. So in addition to our MCR8 and MCR9 genes, which I highlighted in both figures here, a range of beta-lactamases were also detected. And the most concerning findings among these were the findings in relation to carbapenemases, so which I've outlined here in both figures. So carbapenemases are genes that can confer resistance to carbapenem antibiotics, which are one of our last line antibiotics. So we have an OXA48 that was detected in the clinical sample, and we also have an OXA48 down here in the MCR9 isolate that was detected in wastewater treatment plant influent. Then we have an NDM as well in the MCR9 isolates that was detected here in a freshwater river sample. So the co-harboring of carbapenemases and MCR genes can significantly compromise treatment options in relation to carbapenem resistant infections. So we also have here 3 genes that I've highlighted that all are associated with conferring resistance to fosfomycin. So fosfomycin is a reserve antibiotic for carbapenem resistant infections also. So this is another concerning finding. So just in relation to the location of the genes, so we used Platon and we ran our short read sequences through this tool, and we were able to predict that all MCR genes were located on plasmids. We haven't yet identified the location of the MCR8 genes, but this analysis is currently ongoing. So as I mentioned, this is just a prediction. We would need to carry out long read sequencing in order to confirm this. So we have carried out long read sequencing on one isolate so far. We do plan on carrying it out on the remaining isolates that we have. So the one that we've carried it out on is an MCR9 positive Enterobacter from the wastewater treatment plant influent. We sequenced this using the MinION sequencer, and it was assembled using a hybrid approach. And overall, 3 circular plasmids were identified. One was our MCR9 carrying plasmid. So through PMLST, we identified this as an INCHI-2 type plasmid. And on the same plasmid that was carrying our MCR9 gene, 10 other antimicrobial resistance genes were detected on this one plasmid. As you can see, I've listed them here, and some of them contain, or some of them are beta-lactamases. So just to conclude, I suppose we detected MCR genes in a variety of different environmental samples, which is therefore highlighting the spread of these genes and the presence of these genes in the environment and to the environment in Ireland. So our findings suggest that releasing untreated wastewater into the environment might also contribute to the dissemination of these genes within the environment, and then in turn might be spread back to humans or animals. We believe that monitoring the environment for MCR genes could also have important applications in detecting unrecognized dissemination in humans and in animals. And we believe as well that genotypic methods are required in order to monitor dissemination of MCR genes in humans, animals, and environment and in the environment. because it's clear from our results that the phenotypic testing does have its limitations, and that MCR genes may be circulating silently and remain undetected unless they are induced by clistin. So I'd just like to thank everyone for listening today, and thanks to everybody for who's involved in this research, and also to the funders of the research. And I'd be happy to take any questions afterwards.
Thank you very much, Niamh. Great presentation. Thank you very much. So if you have any additional questions, there are already some questions in, but if you have additional questions for the presenters, please use the Q&A function at the bottom of the Zoom screen here, and you can ask them directly. Okay, so there's a long list of questions here for everyone. So well, Niamh, since you were the last semester, since you're already here, I'll just ask, I have one question for you. So you've mentioned something that's very intriguing, that the MCR9 isolates that the were susceptible to colistin, right? But you've mentioned that expression could be induced. So how could it be induced? I mean, what's the mechanism?
Yeah, so all of our MCR9 isolates, as I mentioned, remain susceptible to clistin. So in a lot of the reports that I have come across to date, many of them that report MCR9 detection also report that it remains susceptible to colistin. However, there are some reports that do report the colistin resistance, or resistance to colistin. And they have mentioned this two-component system called QSEBC. Now, I haven't looked into too much as of yet. I'm still looking into our findings and the literature out there on it. But it seems that this two-component system has the ability to induce expression of the MCR9 gene. So in studies where it has been found, they've also found that the isolates are clistin resistant. But there was one report that I did come across that kind of contradicts it a little bit, because they found an isolate where the MCR9, an MCR9 positive isolate that had this two-component system, but also remained susceptible. So they believe that there are other molecules or genes or factors that can express the MCR9 gene, but further studies, I think, are really required in that area. It seems from the literature that I've read to date.
Okay, yeah, brilliant. And were these MCR genes found in a plasmid, or were they chromosomal?
Plasmid, yeah,
plasmid,
yeah.
right.
So usually the two-component system is found up or downstream from the MCR9 gene on the plasmid.
Okay, and I suppose colistin is very relevant nowadays, with a huge rise in carbapenem resistance. Kind of remains one of the few viable options, and it just came back from the dead really after due to its high toxicity for years wasn't used, but now it's kind of last resort. So seeing resistance circulating in the environment for colistin is highly worrying, and of course we don't know what it means for the population, unfortunately. Okay, thank you very much, Niamh. Adrian, I don't know if, can you hear me?
Yes, please, I can.
Okay, so I have a few questions here that are coming in for you. So one question was, could you you've showcased very nicely how the populations, the microbial populations, differed between the urban and the peri-urban or rural cohorts. Could you separate the populations based on their resistome profiles? So not the microbiome profiles, but if you were to look only at the resistome, would you separate the two populations? Did you see differences there?
Yes, that is specific not to the entire resistome, but to resistance genes that belong to a certain classes. Within those, you can see that the urban carries just a certain profile, and the rural setting a certain profile of resistance. And that, I think that was true for the macrolides and aminoglycosides. Okay. So you can for some specific antibiotics.
And are these the antibiotics that are primarily used, let's say, for farming and
this would be for aminoglycosides and macrolides, and macrolides, specifically tylosin, is highly used in pigs. So that would really concur with what we see in the resistome.
Okay, that's very, very interesting. And could you see, so in the urban population, you've mentioned that the resistome was richer in a way, there were more genes and more populous. So is there any explanation or any ideas that you may have? Why could that be the case in the urban population?
So in this study, we used the, I'll go back a little bit to the quantification of genes using qPCR. We used the gene load as our sort of proxy to selection pressure. Mm-hmm. If we imagine that if an environment is using a lot of antibiotics, we'd expect that would sort of push up the number of genes and the diversity of, well, the number of copy number of genes that we would see in that environment. And we see that in the urban, really, we have a higher gene load in the urban settings than in the rural settings. And also the diversity when it comes to the resistome of genes. Some of the figures I didn't show, but we do see that you have a higher diversity of genes in the urban than the rural setting. And we think this is primarily driven by the use of antibiotics in those settings, which is sort of the hypothesis we set out really to investigate.
Yeah, of course. Of course. And Adrian, based on that's another question here, based on the data you've accumulated, you've mentioned you did some shotgun metagenomics as well. Is that correct? To a kind of sub cohort, I suppose, due to the expense of the technique and were there in particular microbial taxa that they were linked with AMR carriage that they kind of popped up in the analysis, some particular species that they were carrying antimicrobial resistance genes more than others?
Mm-hmm. So we did what they call it's called MAGs, a metagenomic assembled genomes, and we assembled about 2,000 genomes, and from that we had this, phylogenetic tree. And so there are specific clusters of taxa that belong to the phylum Proteobacteria that seem to carry a significant, a high diversity of resistance genes to a number of classes of antibiotics. So we see that with Proteobacteria, but we also see a bit of that with the Actinobacteria. So those two. I didn't show that figure, but we've done this metagenomic assembly of genomes.
Okay. That's very interesting. Great. Wow. Okay, so I'll have, the time is kind of pressing a little bit, but there's so many questions. So Adrian, one last question for you before I go over to Alex. So you showcased the thing. It was an excellent example of how you could use omics plus big data in a way, and to kind of get this, because I think the relationship between whether it's the environment or animals and human health is just so complex, and the dynamics are so subtle that you really need everything that you can get to be able to pinpoint it. What's the next plan? What comes next from this? I mean, are you thinking of maybe using like metatranscriptomics or any other method to complement what you've already have there, or what's the next step?
So we actually do have RNA stored also that was extracted from this. So if we do get money, we can do metatranscriptomics. It just has to be a specific question we want to answer. I think for me, this study just, I wanted to look at the complexity and not really come up with because most people would like to ask, okay, so now that you have shown this, tell me what whether the farmer is transmitting to the pig or the pig is transmitting to the farmer. I think by demonstrating that the fundamental architecture that drives what we see is really the same and at the same scale in pig and farmer, I think from that we can infer whether or not there's transmission between farmer and pig And that's really relevant with regards to how we design intervention that target animals, because if we use a broad, really use very hard interventions, they fundamentally affect how farmers really do their agriculture, because antibiotics are really critical in food production. So I think for me, the work is really to demonstrate that the fundamental architecture of how this happens is similar. There are things that we can exploit. And from that, we really think that there's transmission going on between farmer and animal.
Of course. Yeah. Okay. Adrian, thank you very, very much. Thank you.
Cheers. Cheers.
Alex, there is a question here for you. You've mentioned that you use porcine fecal samples. Did you test any other type of fecal samples besides porcine for your WorldCom study?
Yeah, we did. So we also looked at bovine, chicken, and alpaca. Our bovine and chicken fecal samples were actually spiked because we didn't have CTXM positive samples of those. And the assay was successful in evaluating those along with our sample prep method for bovine and chicken. The alpaca was CTXM positive from our friends in Germany who sent them over. However, the consistency of alpaca fecal samples was a bit nitty-gritty. And so I think our extraction method was a bit too simple for for our alpaca samples, but we have tested them on several other fecal samples besides pig just to evaluate the assay.
Okay, yeah. Okay, brilliant. Thank you very much, Alex.
Yeah.
Thank you. I'm aware of the time is 10:30 now, so we can leave it here, I think. I would like to thank you all very, very much for your presentations. Adrian, Alexandra, Neve, and Maeve, thank you very much. Great work, great presentations. And thank you all for joining the session today. In the morning, please stay around. We'll have a 15-minute break, and then we're moving on to the emerging pollutants theme of the conference. So there's a lot of great presentations lined up for you. So thank you all for joining, and thank you all for participating. And we'll see you after the break. Thank you.